All right, so what we're going to do is we're going to do some practice with replication, transcription, and translation as basically a review of everything we've gone through. A couple things that we've got to keep in mind is that DNA is a double helix. Don't forget that this green portion that's on the outside here and here, that is going to be your sugar as well as your acid, and then inside we have those nitrogen bases. So first thing we've got to remember is replication. If DNA is going to make a copy of itself, what's the other side of the DNA going to look like? It's going to open up in the middle here, which we'll get to some images of that in a second, where it opens up, and then those nitrogen bases are going to be coming in. So first thing you have to remember is what are those bases that match up with these. So take a second, just look at them, see if you can match up what base would match with T there, what would match with C, what would match with G, and so on. Okay, so after taking a look at that, you should have matched these. Double check, make sure your A's match up with T's and your G's match up with C's. Here's another example for you, again, making sure your A's and T's match up. Run through them real quick for yourself. And you should have gotten this as an answer. And there's a few more practice on here for you. Just remembering A matches with T, G matches with C. So when we're doing replication, a couple other things that we want to remember is that this is going to happen during the S phase of mitosis. And that means that the DNA makes a copy, so it's going to have 200% of the original. And then that way, each of the two new cells that are produced in mitosis will each get 100% of the DNA, and it'll be an exact copy. All right, so the reason that we have to make RNA is going to be because that DNA is stuck in the nucleus. So I'm only showing one side of the DNA here in green. So this is your DNA template. Okay, the DNA is going to open up just like it does in replication, and the one side is going to serve as a template or a guide. So since that DNA is stuck and the proteins are made out in the cytoplasm, we're going to make what is called messenger RNA. We're going to send the message out of the nucleus by making this. This first step is going to be called transcription. And we have to remember that RNA is a little bit different. It's only single-stranded. So we can see we've got just this one strand that's going to be made. It's got a different sugar, and T is replaced by U. So that means that anytime we would normally put a T, for example, this very first one, we would normally put a T here, but there is no thymine in RNA. So we're going to put a U there. So take a minute, see if you can figure out what the pieces right in here would say. So here is what your answer should have looked like. Double check. Here's another practice one for you. See if you can figure out what would go on the RNA, on your messenger RNA that's going to go out to the cytoplasm to make our protein. Again, this is transcription, so what would that messenger RNA look like? Here's what your answer should look like. There is one typo that I kind of wrote over with a U right there. No T's in messenger RNA. Another practice for you. Take a second, pause it if you need to, and then check your answer. Here's what it should look like. And another practice for you. Again, pause it, check your answer. here's what your answer should look like. All right, so I'm going to try something a little new here. We are in the next step, which is going to be called translation. And with translation, this is where our messenger RNA, which I have right down here, is going to move out into the cytoplasm. And because that's where our ribosomes are, and that's where the protein is going to be made. So on this orange strip down here, we've got our messenger RNA. And these guys that are up here at the top of my screen, those are going to be the transfer RNAs. And the job of the transfer RNA is to take the amino acid, and they have a code down here. And it's going to pair up the same way that it does with the messenger RNA to make DNA. It's going to have the same kind of pairing rules. So, for example, if I've got A here, we have to match that with U. Remember, RNA does not have any sort of thymine in it. So that means that U here would match with A, and G here would match with C. So this set of 3 letters right here on the messenger RNA, that's going to be called a codon. And the tRNA that's up here is going to, the one that matches, the one that has UAC is this one with the dark green. This guy's set of matching here, he's called an anticodon. And he's going to go ahead and he's going to bring his piece over here to match. And he's carrying an amino acid. So these little dots right up here, those are going to be the amino acids that they bring in order to build a protein. All right, so continuing on with our process of translation, again, we've got our messenger RNA down here, just like I said in the previous slide. We've got our tRNA matching its anticodon with the codon that's on the mRNA, and it brings the green one. So our next step then is to bring the next piece. So A matches with U, A matches with U, and G matches with C. And so that's going to bring this pink one right here, and those amino acids are going to join together, they're going to connect to each other to start building that protein. Okay, so now we've got our 2 amino acids. This guy is actually, this transfer RNA, he's going to leave and he's going to go get a new amino acid so he can do his job again. And if we look here at our next codon, we would have GUA. So that means the orange one is going to come in next. So now I've got the orange, or the green and the pink here attached to each other. The orange is going to attach, and then for this messenger RNA sequence, the last one, anticodon on my transfer RNA that's got to come in, is going to be this next one here. So this final slide here shows us how we've got those amino acids connecting to each other, and these amino acids then here are going to be your protein. So just a real quick recap, this is happening in the cytoplasm. This is going to be the process of translation, and this is going to be where our messenger RNA has codons that are going to match up with our anticodons that are on the transfer RNA, which bring the amino acids to build the protein. That's it. So how do we know which amino acid is going to be brought? So what we're going to be looking at is what is called a codon wheel. And so with the codon wheel, we're going to read it from the inside out. So if my codon on my messenger RNA, because that's where the codons are, says A, then the amino acid that's brought is called lysine. If my code said maybe CAA, then it would be glutamine. So we're going to do some practice with the codon wheel. You can see that this image right here, we have messenger RNA at the bottom. We're going to use our codon wheel to figure out what amino acid is brought. So UUG is going to be bringing us leucine. AAG brings lysine, so AAG, that's that one right there, lysine. CAU brings histidine, and then GAC A, and C brings aspartic acid. So we're going to do some practice next. All right, so here's our codon wheel. Down at the bottom, we have our messenger RNA right here. I want you to take a second, pause the video, and see if you can figure out what all the amino acids are that would be coded by this messenger RNA. So this is the answer you should have gotten. All right, so here is another practice for you. Pause the video. See if you can figure out what the amino acid sequence would look like. And then pause it now. And then you can go ahead and check your answer. All right, so since I'm new at this, look who I got to join me on this one.
Hi.
Hey, so you got one more practice here. Remember, practice is going to make perfect. So you've got your messenger RNA. Go ahead, use your codon wheel, and pause the video. We'll come back to you. Pause it. They have to pause it. And then check your answer.